Welcome to a new series at Caltech. Lee Hood is our inaugural Presidential Distinguished Speaker, launching a new series given by scholars and practitioners grappling with the most compelling intellectual pursuits and profound societal challenges. A member of all three national academies, the recipient of significant honors including the Lasker Award, the Kyoto Prize, the National Medal of Science, and the Caltech Distinguished Alumnus Award, Lee Hood's career has been marked by vision and the means to achieve it. His Caltech education, BS 1960 and a PhD in 1968, bookending his MD from Johns Hopkins, and his postdoctoral appointment at the NIH, all served as foundational experiences in his quest to connect scientific discovery and technological innovation with clinical advances. Equally comfortable in the laboratory, in the classroom, in the hospital, and now in the boardroom, Lee summoned this wealth of experience to herald a new era in systems biology, an approach to biological research that translates complexity for direct use in both basic science and disease treatment for the benefit of us all. In Lee's own words, recalling advice from his mentor, Bill Dreyer, that led to his enrollment in the PhD program at Caltech, if you want to practice biology, do it at the leading edge. It's more fun there. Don't be a librarian that's just doing in more detail what someone else has already done. 2, if you want to really transform a field, you better do technology that gives you a new view of the biology. So those were the core of what I started at Caltech: molecular immunology for at the frontiers and technology development for opening up new fields, new vistas. Lee took this as his inspiration for his doctoral research and for his development as a Caltech faculty member from the 1970s to the early 1990s of the gas phase protein sequencer, the DNA synthesizer, the peptide synthesizer, and the automated DNA sequencer. The invention of the DNA sequencer in 1986 made possible the Human Genome Project and the embrace in biology of big science as a means of parsing the human genome and in marshaling the personnel and computational power necessary to map the genome and ultimately gain insight into chronic disease. Lee moved to the University of Washington in 1992, founding a new department of molecular biotechnology with a focus on systems biology. In 2000, he launched the Independent Institute for Systems Biology with the aim of creating a multidisciplinary research culture from the ground up, inverting the traditional focus on reductionism in biology toward integration. Today, Lee will tell us about applying systems biology to healthcare and advocating P4, where patients can enjoy a medical approach that is predictive, personalized, preventative, and participatory. It's my great pleasure to turn the podium over to Lee Hood.
Well, thank you very much. As you can tell, I was here at Caltech for some 30 years, and I'll say it was a wonderful place to grow up scientifically. And it led me to first start thinking seriously about data-driven healthcare, which is what I'm going to talk about today. Phenome Health is a nonprofit organization I created about a year and a half ago to drive forward this idea of catalyzing a paradigm shift in healthcare from disease care to wellness and prevention care. And I'll divide my talk into 3 parts. Number 1, I'll give you a historical and contextual background of how I think about data-driven medicine. Number two, I'm going to actually draw 6 stories from the data we generated from a cohort of 5,000 people over 4 years that point to the kinds of things that data-driven medicine can do. And finally, I'll tell you how we're actually going to get there in the future. I was, as I said, interested in 1970 when I came to Caltech in human complexity. I'd really decided I wanted to study human biology and human disease, and of course was stopped dead by the enormous complexity of the system. What was clear to me at that time was, one, you deal with complexity by using a lot of data, and two, a lot of data didn't take you very far unless you could coherently organize it. And these were 2 principles that set me off in exploring, often with others, a series of 7 paradigm changes, each of which opened up new ways to approach human biological complexity, but at the same time framed how I thought about biological complexity. The first of these was bringing, in a sense, engineering to biology with the invention of 6 instruments that automated the analysis of DNA and proteins in various ways. And this opened up the possibility of generating large amounts of data on human beings. One of those instruments, the automated DNA sequencer, got me invited in 1985 to the first meeting ever held on the Human Genome Project, where 12 of us voted unanimously it could be done, but were split 6-6 on whether it should be done. Those against worried that big science would swallow small science. And of course, the Genome Project did get done, and from my point of view, the most important thing it did was to give us access to human genetic variability and our ability to correlate it with both wellness and with disease phenotypes, and we'll talk more about that at a later point in time. In '92, with the help of Bill Gates, I moved to the University of Washington to start the first cross-disciplinary department of molecular biotechnology, and it was enormously successful. Writing the software, key software for the Genome Project, developing the first 2 critical techniques for the newly emerging field of proteomics. And we invented there the inkjet technology for the rapid synthesis of DNA that Agilent still uses yet today. But it was clear that if I was to impose systems biology on top of that cross-disciplinary platform, it would have to be done in another context, and that's when I resigned from the University of Washington in 2000 to start the Institute of Systems Biology. And of course, systems thinking is global and holistic, and it's dynamical, and it puts together all of the elements that one needs to assess human complexity in a variety of interesting ways. And in fact, pointing the thought of systems approaches toward healthcare, it became obvious that healthcare ideally should be predictive, preventive, personalized, and participatory, and that it fell in 2 major categories: wellness on the one hand, disease on the other hand. And at that time, the healthcare of 2000 was about 99% disease care and almost no wellness care. That all began to change in 2013 and 14 when we took on a 100-person project, and then later when we started a company called Aerovale that took on a 5,000-person project over 4 years, where we did genome, phenome analyses, and I'll explain those in just a minute. But that generated an enormous amount of data that we've continued to analyze up to this point in time. And it led us to this idea of precision population health, generating lots of data on each individual in a population and using that data uniquely to specify how to help optimize the individual's health. And the final paradigm change was to realize that the 5,000 people we developed with AirVale was way too small to convince the medical world about the power of personalized precision medicine, and that we would need a 1 million-person project over a 10-year period with this genome-phenome analyses in order to prove two critical things. One, we could improve enormously the quality of health care for that cohort. And two, we could affect enormous savings in our $4 trillion healthcare costs and so forth. So these set a framework then for thinking about a systems approach to generate big data on individual humans. And the real key is to realize that blood is a window into health and disease. And it is so because every organ in your body sheds molecules into the blood. And if you can learn to read the identity and quantify those molecules, you can indirectly assess the status of that organ. And that's precisely what we've done in direct and indirect ways over the period of time. Of course, converting the data we generate into digital form, and then ultimately transferring that data into the biological networks that underpin physiology and disease. And thus we can visualize and recreate, in some sense, the two and even three-dimensionality of human complexity, and we'll see how we do this in just a few minutes. Now, what exactly do we mean by genome and phenome? The genome, of course, is the basic set of chromosomes present in every cell in your body, 3 billion DNA bases present in that genome. It's an enormously complex structure. To a first approximation, unchanging itself across developmental time. Whereas the phenome, on the other hand, is a description of you at many different stages of your life. In fact, we all constitute an infinity of phenomes, and the three influences on what your phenome is at a given instance in time is your genome, it's your behavior, and it's your environment. And of course, we can begin to measure the phenome by looking at blood analytes, by looking at the gut microbiome, by looking at digital health signals from the brain and the Fitbit and things like this. And they collectively constitute then a data cloud for each individual that increases longitudinally with time as the assays go on. And again, we built on the population at AirVale, 5,000 over a 4-year period of time. And this led to a fascinating vision of what we really want to do. And that is, we now had the ability to assess and optimize the health trajectory of each individual in a population. And of course, the health trajectory has 3 different phases. Most of us start in wellness. Almost all of us transition at 1 or more times to disease. The disease progresses and, in most cases, terminates itself, and we return back to wellness and so forth. Now, what's interesting about these phases is in the mouse, we've looked at 2 model systems, one in a neurodegenerative disease and the second in a neurodegenerative cancer, and been able to demonstrate that the biological networks that are disease perturbed increase in an exponential manner as you go from the point of transition all the way up to a clinically diagnosable disease. And that means, obviously, that you want to be able to reverse disease at that earliest transition point. And so these 3 points, wellness, transition, and disease, are what we'll focus our systems approach and big data analysis on. And the 3 phases then, interestingly, lead us to very interesting conclusions. One, most of you, if you're well now, may represent 30% of your potential wellness. And with the actionable possibilities that come from a data-driven analysis of your health trajectory, we can create unique sets of optionable possibilities that will improve wellness. And part of the question is, how well do you want to be, and how much are you willing to commit to participating in this process? With regard to transitions, we have the ability to think about how to reverse disease in a relatively simple fashion long before it ever manifests itself in the future, and we'll return more to that. And then, of course, with regard to disease itself, we'll use the approaches of systems biology. And let me just say, for most chronic diseases, there are 2 really critical questions we'll return to. For diabetes, how many different subtypes are there? And the answer is probably 4 to 5. And second, for each subtype, what are the nature of the state transitions that mark the most signal features of diabetes in that particular subtype? And I'll tell you exactly how we'll go after both of these questions in some detail. And I would argue that this science of wellness and prevention is going to be the biggest revolution in the history of medicine once it actually happens. So how do we go about then proving the principles of genome, phenome, data-driven medicine? One, Airvale gave us 5,000 people, and those data are going to lead, as you'll see in a moment, to 6 different windows on what it is to be healthy. Number 2, we will start in the future collaborating with a company that specializes in digital analysis of brain health trajectories and the ability to assess and optimize cognitive functions that are lost, and to assess them and optimize them even out into one's 80s. So the brain is plastic until the end of your life if you take proper care of it and so forth. And finally, of course, is the whole question of the gut microbiome, in a sense, a 3rd major organ in your body that contributes in enormous ways to your health. And we'll talk about several of those ways that have come out beautifully from the papers that we published, and here are 24 of the papers. There are almost 30 that have come from the Airvale data. And as I said, what they each give us are different views of what health can be in the future. And so story number 1 is talking about how we can, in principle, prevent chronic disease. And one of the striking observations we made in the course of the Airvale population was a woman that was diagnosed with stage 4 pancreatic cancer, the worst form of pancreatic cancer, and she was called Eve. And what we decided to do was to look at the bloods that were drawn in the 2-year period prior to her clinical diagnosis and ask if there were perturbations in those bloods of molecules that could reflect that disease. And of course, the answer to that question was there were. It turned out to be 5 of them, and one of them that we've diagrammed here to a first approximation. As you get closer and closer to the disease, the quantity of that protein gets further and further away from the normal of the rest of the population that existed at that point in time. 3 of those 5 proteins mapped into disease-perturbed networks that were unique to stage 4 pancreatic cancer, thus giving us a firm diagnostic conclusion as to what it was about. Now, the other thing we did is we had 167 transitions from wellness to disease all told in that population. 35 of them transferred to cancer. We looked at 10 different cancer examples, and in all 10 examples, we saw exactly the same thing that we'd seen here with Eve. And in fact, with the million-person project, our estimate is we'll see 200,000 transitions. And this means for the transition to both common and relatively infrequent chronic diseases, we'll have irrevocable markers that will tell us what disease we're transferring to, and we'll begin to specify the nature of the therapeutic early reversal that will lead in time to an enormous diminishment of chronic diseases. So I think this is really a significant case. Story number 2 is scientific wellness and actionable possibilities. And here, basically, we take the data of 108 individuals, the first population we looked at, and that includes proteins and metabolites and clinical chemistries and the gut microbiome and digital activity and whole genome sequence. So it is an enormous amount of data. And we used pretty classical statistical methods to identify 3,500 statistical correlations between data bits in one of the 6 data types with data bits in the other. And what is important about those correlations is where it made sense, they led us to the literature, and the literature explained the nature of the actionable possibility those statistical correlations represented, and where appropriate, that could then be applied back to patients. I'll say in another study we did, we identified and quantified 5,000 proteins, not 500 proteins, and showed that moved the statistical correlations to 35,000, and that gave us even denser views of correlations, actionable possibilities, and actually an expansion of the size and interactions of the corresponding networks and so forth. So what we're in a unique position, I think, to do with this is with the new million-person project, we suggest there'll be at least 10,000 different actionable possibilities, and it will be essential for AI to be able to deliver them each to physicians and their patients, saying what the nature of the actionable possibility is, what the action should be, and justify it in terms of scientific validation. And we should easily be able to do these kinds of things. There are 2 complementary kinds of wellness. There's classic wellness, which is diet, exercise, stress, sleep, and so forth. And of course, there's the data-driven wellness that we've talked about. And I'll just say that actionable possibilities come from many aspects of the genome, and actually thousands of actionable possibilities come from there. And one of the most interesting for many is there are hundreds of variants that correlate with various athletic injuries. And if you know you have such a variant, in most cases there are things you can do to avoid an ACL tear or groin injuries or simple things like that. There are many actionable possibilities in the phenome side of things, optimizing vitamins and supplements and so forth, being able to look at these early chronic disease transition points and reverse them in time and so forth, and biological aging and metabolic BMIs, which we'll talk about in just a moment. But to give you a concrete example from myself about an actionable possibility, I was extremely low in vitamin D, and that's essential because it predisposes you to a variety of diseases, including cardiovascular disease, including dementias and other things like that. I found that 1,000 international units, which usually works fine to elevate your levels, didn't touch me. And it turned out that I had 2 variants that blocked the uptake of vitamin D that only could be dealt with by using 15,000 international units to bring my level back to normal and 5,000 to maintain it. So putting together genetic data with blood data allowed you to do an integration that led to a really important actionable possibility. But the other point that's really important is data-driven approaches also individualize the classic wellness in the sense that each of you can optimize your exercise, can optimize your diet, can optimize sleep, and a variety of things in ways that are different from many of your other counterparts. So the data-driven wellness brings individuality to the traditional wellness itself. So story number 3 is genetic risks are essential to learn, and we now have at least 150 conditions for which we can identify for you your genetic risk if we have your complete genome sequence. And the other point I'm going to make is low-risk and high-risk people will be treated differently in the future. So the genome, of course, I've already defined, and the variants in the genome that carry out either physiologic traits that encode physiologic traits or disease traits can be identified by large-scale studies of hundreds of thousands of individuals. And to give you an example, height is perhaps the most best-studied phenotype for genetic traits, with 180,000 making up about 60% of what it takes to determine what your height is. And for many of the other chronic diseases and so forth, we can put together a collection of variants that each contribute in a small way to that genetic predisposition and call that a polygenic score. And if you have then the whole genome sequence, you can divide people into those that have either high risks or low risks for that particular genetic trait. And here's an example, actually, of a whole series of individuals in 4 different diseases. And you can see with increasing genetic risk, the disease phenotype goes up enormously. And what this slide says unequivocally is if you're in the upper 5% of a disease risk type, you ought to be treated to a first approximation like you have the disease. And if you do it in a preventive manner, you may be able to avoid the disease. But even more striking was the one example we studied in enormous detail, the genetic trait for predisposing to high LDL cholesterol, which is shown here in the Aravale population. So LDL cholesterol increases linearly with people of increasing genetic risk in those that aren't using statins or other similar kinds of chemicals. And what we were able to demonstrate beautifully is if you're at high genetic risk and have high cholesterol, the only thing that will bring it down are statins or something similar. In contrast, if you're at low genetic risk and have high cholesterol, diet and exercise and the like bring it down beautifully. And the really important point is that that high and low genetic risk individuals have to be treated differently, and we think this is going to turn out to be true for a lot of diseases. We can readily compute now your genetic risk for more than 150 diseases, and I think this is going to be one key feature of data-driven health in the future. Story 4 is really an interesting one because one of the questions you have to ask yourself is what's a metric for healthy aging and extending your health span? And we found one in the aerophile population in that we were able to show as people got older, their ability to tightly confine the expression of their blood analytes, proteins and microbiome and clinical chemistries was lost. And we could use that to create an algorithm that gave us the ability to determine one's biological age, the age your body says you are rather than the age your birthday says you are. And the basic idea is the lower your biological age is relative to your chronologic age, the healthier you're aging. And what we were able to demonstrate beautifully is that we could do this calculation with proteins, with metabolites, or with clinical chemistries, any of the 3 blood elements. But the one that worked best, for a reason I'll tell you in just a moment, was the one with metabolites. And the second point I would make is for every year that individuals in the aerophile population stayed in the program and practiced their actionable possibilities, they lost a year to a year and a half of biological age. So one of my friends lost up to 10 years of biological age, and that raises interesting questions about how low you can elevate your biological age. I will say my biological age is 15 years younger than my chronologic age. So it means you can start thinking about retirement and things like this in a very different way, using your biological age and so forth. What I think is really interesting is that the lowered biological age was seen in healthy people, as we've just described, but we had a whole series of people that had various diseases, most of them trivial, but in every single case, the biological age was well above the chronologic age for sick people. And for diabetes, it was actually 6 years above the biological age. And I think the other thing that was really fascinating is because we used metabolites, we could actually determine the biological age of major organs like the liver, the kidney, your immune system, your lipid elements and things like that, and compare it with your overall chronologic age, and you can actually specifically address how to make your kidney, which is maybe had too high a biological age, come back to a more normal, reasonable level. So the metabolites gave us the ability for the individual to determine how to decrease their biological age. And there are a number of methods out there for doing biological age. The metabolic approach is the only one that gives you specific actions that you can do. The final point I'd leave is age is a major factor in correlating with all chronic diseases. And of course, the interesting question is, how much does society save by slowing down the aging process? And there are people that have argued that it can be multi-trillions of dollars, which is a staggering sum for doing this whole process down. And of course, the other thing that slowing down your aging process means is that your health span begins to match your lifespan. Your lifespan is how long you live, but for most people, the last 20 years can be fairly miserable. Your health span is how long you live in a healthy manner and fashion. And we hope with this new data-driven health that we'll see people in their 90s and their hundreds, mentally alert and physically capable and the like. The metabolic BMI is absolutely a transformational kind of observation. The classic one, which uses height and weight as a means of measuring its indices, and it correlates with chronic disease and mortality and a variety of things like that. But it miscalls things 30% of the time. For example, muscular people are often registered as obese. So athletes often end up being obese. And it has no capacity to respond to short-term changes in metabolism. Whereas we've learned now and trained an algorithm for metabolic BMI that is able to correct all of those deficiencies. It's accurate, it responds to change. It correctly calls the difference between athletes and obese individuals and the like. And I would predict in a paper published in Nature Medicine just a few weeks ago that this will come in a very short period of time to replace the classical BMI. And it's a beautiful way of overall assessing regularly metabolism of the human body. And the last story is the gut microbiome in health. And of course, one paper that we published some years ago showed that we can actually determine the diversity of the gut microbiome from looking at 11 blood analytes, 11 metabolites, and so forth. And greater diversity in the gut is equal to greater health, and so forth. A second thing that we did was showed that the gut microbiome had a profound impact on how statins work. And let me just say, to cut to the chase, statins do two important things. One, they cut your LDL cholesterol down, and two, they, with a regular frequency, cause type 2 diabetes. And I can just say that we could get a microbiome composition that optimized cutting down LDL while minimized the diabetes. And there were other microbiome constitutions that did exactly the inverse. And we'll be looking at most drugs in this context. The gut microbiome may be one of the most important determinants in that regard. And an absolutely fascinating observation we made a couple of years ago was to look at people from 60 to 90 and to assess their gut microbiomes. And what we were able to demonstrate beautifully is if you aged in a healthy way, two things happened to your gut microbiome. You lost the core microbiota that you had at a younger period in life. And as you aged, you became more and more uniquely differentiated to a unique microbiota. Now, in contrast, people that aged poorly neither lost the core microbiota, nor did they age in a manner that led to unique differentiation. And what was interesting is we showed that people in their 80s that were healthy as opposed to unhealthy, the unhealthy had 4 times the rate of death in their 80s that the healthy had. So this says we're going to be able to design in time microbiota which will be consistent with long and healthy lives and so forth. So what's next? The Human Phenome Initiative, that is this idea of a million-person project funded by the government, Second Genome Project, over a 10-year period of time. But in order to get started immediately, because that'll cost a lot of money and require a lot of politicking, in the short term, what we're going to do is use our data-rich approach to each of the 4 major chronic diseases, starting with type 2 diabetes in a way I'll describe in just a second. And of course, the long term is to do the million-person project and prove unequivocally an increased quality of healthcare and a decreased quality in the cost of healthcare and everything. So where is Phenome Health right now? Well, we've actually created a clinical partner called Guardian Research Network that has access to 50 million patients in 13 different eastern and southeastern states. And they've learned to extract from those patients their electronic health records. And we'll return to that in a moment. Posit is the company that we're collaborating with on digital brain health and its optimization. Technicity you use to build a unique multi-omic platform that is compatible with all three types of US clouds and so forth. And Google, we're collaborating on to use hyperscale AI. ChatGPT is one example of that kind of thing, to be able to analyze patient records with a power, dimensionality, and speed has never been conceived before. ISB, we're collaborating with people that are expert in knowledge graphs and people expert in digital twins, and we're focusing all of those on creating a digital twin model of diabetes that we'll use in the first project. Scientific American has created a 75-page article on the new science of wellness, which I think is a marvelous discussion of where we're going in the future with all of this. We've started Phenome Health based on philanthropy initially, and we expect $25 million to get the diabetes project started at the beginning of the third quarter. Now, let me just show you how important racial diversity is in this data-driven health. This plot shows individuals that are homozygous for ApoE4, the highest variant causing Alzheimer's disease. And what it shows is Japanese are 3 times as likely to get it with that double homozygote as Caucasians are, and Hispanics essentially aren't affected by all. And this is going to be true of many different diseases, so we need racial diversity to be able to accurately call the diseases and create appropriate treatments. And Guardian Research Network, again, lies across large African American and Latino populations, and we plan to recruit them according to population size. The profiling of the human phenome that we'll do in the million-person project will have more than 10 times as much data as we had before, and it will have many different types of data we didn't have before, including electronic health records, including the brain analyses and so forth, and including these data-driven metrics, which are biological age and metabolic BMI. So what's next on the Human Phenome Initiative? In the short term, again, it's going to be applying our data-rich approach to type 2 diabetes. And this vertical that you see here shows the 4 major types of chronic diseases and which of those we'll be studying in the future. We've already fully planned the diabetes project. We hope to, in the next few months, get funding for it. And we're well along in planning and getting funding for the cancer project as well. But the idea is really an interesting idea that will answer the 2 questions I raised earlier. How many different subtypes does diabetes have, of subtypes of diabetes are there? And we think it'll be 4 to 5. And within each of those subtypes, what are the state transitions that mark signal events? For example, one we're very interested in is a state transition that goes from diagnosed diabetes to chronic kidney disease, which is one of the most costly complications in all of healthcare today, and we'd like to be able to identify and reverse people, let them avoid getting that in the future. So the approach that we're going to take is we've already studied 10 million people from the normal population. 11% of them were diagnosed diabetics, 30% were presumably pre-diabetics, and the rest were normal. And we're looking for blood clinical markers that will allow us, 1, to determine subtypes, and 2, to determine transitions in substates. Once we have these markers, we plan to select roughly 250 patients for each, say, of the major 4 subtypes, and then follow them through their interesting transitions for a period of 4 to 5 years. And summing up that knowledge, will be in a unique position to have clinical trials that intervene for drug targets and biomarkers and so forth. But with this data-driven approach, in a period of 5 years, I think we'll learn more about diabetes than we have, frankly, in the last 50 years. And of course, what it means for diabetes is individuals that will respond to one type 2 diabetes drug ineffectively can be divided into the 4 subclasses, and we can generate drugs for each of those 4 different subclasses. And of course, this raises an interesting problem of how do we generate all the drugs we're going to need to do all the things I've talked about. And let me say that we're thinking about starting a new pharma company that has a strategy for generating peptide drugs that has scale, namely we'll be able to generate 10 to the 12th, and we'll be able to select them either at the level of the cell or at the level of the protein really effectively. So I think that's going to open up possibilities for the future. But if we subtype diabetes, we get drugs for it. Diabetes is one of the major cost effectors in chronic disease for contemporary medicine. We spend 86.6% of our dollars, the $4 trillion we spend on chronic diseases. Suppose we could cut those in half in a 10-year period, could have a dramatic impact. And the same is true of drugs. The 10 most popular drugs, on average, 1 in 10 people respond really effectively. And that varies from 1 in 4 to 1 in 25. What we'll be able to do with the data-driven Phenome Initiative is within a few years, we'll have biomarkers that will show for all the major drugs exactly who can respond and who can't. And that, in principle, could be a savings of 90% of $600 billion. So these things lead to large sums of money. What's next? The Human Phenome Initiative, the million-person project. And basically what we think it can do is take care of the 6 contemporary challenges of healthcare. Improve the quality of healthcare through scientific wellness and many of the other things I've talked about. Deal with the aging population by this aging metric and its ability to bring down the aging process itself. The explosion of chronic diseases, if we can reverse them, as I've talked about, or if we can define the subtypes and get drugs to deal with them, either of those would begin to approach it. The diversity, equity, and inclusion, of course, is a consequence of our partner in the South being able to recruit from populations they encompass. And finally, exponentially increasing costs and so forth are something that can be handled in several of the ways I've talked about here. So the final thing that I'll talk about then is saying a systems approach to healthcare is P4, as I've talked about. And what's interesting about P4 is the first 3 Ps are science, and I hope I've begun to convince you we really can manage that part of it. The 4th P is sociology and economics and education and all these other things. How do we persuade the patient, the physician, the healthcare leader, the healthcare regulator, the healthcare technologists to change a whole system from profit focus on disease to a profit focus on wellness and prevention? And I would argue the basic idea is to be able to show them that wellness and prevention will save them trillions of dollars in healthcare in the future. And that's certainly what we hope to be able to do with the project. I will say, and I know it's getting late now, so I won't go through all of these, we've done an enormous amount on education And I think education is really critical for this project and the fourth P, quite frankly. Written a textbook on systems biology and systems medicine that'll really be great for medical school students. We've created a high school senior class based on the systems medicine chapters in our textbook. 20 modules. These kids, when they come out of high school, will know more about where medicine is going in the future than 95% of the doctors that are out there. We've written a book, The Age of Scientific Wellness, and I'll show you that in just a second. And we're starting a 90-minute documentary on wellness over the last 5,000 years entitled The Phenome Age, A Quest for Wellness and so forth. So what are the benefits of data-driven health? We're going to shift from disease to wellness and prevention. We're going to democratize healthcare so that everybody will have a chance at data-driven wellness, and that will require the development of technology whose ultimate response is going to be to have a tricorder you can use at home that will make all the measurements you need on you at appropriate periods of time. And finally, I think we'll have a third industrial revolution where AI will deliver the thousands of actionable possibilities and unique combinations to each of the individuals as they frankly need them. And I'll close by saying this book just came out from Harvard University Press, and it has everything I've talked about here and a lot more if you're interested in following up on it, and you can get it from Amazon Press. So with that, I'm be glad to answer questions if there are questions. Okay.
Hey.
There are mics set up on either side here, so if you have questions.
Great. Ryan Sinnett, class of 2007. I got a couple, actually. The quick one is, how many pull-ups can you do? How many pull-ups can you do?
How many
chin-ups
can I do? I can do 5 pull-ups, but I can do 200 push-ups.
That's a lot more than me. I think the question I really wanted to ask...
Where did that question come from?
Sorry? Well, you said you're 15 years younger than you look, and
yeah.
You look pretty young, so I figured probably do a few pull-ups. You talked a lot about increasing health span and trying to make it go out toward end of life, toward end of lifespan. And I think you talk about these different measures, and there's a bunch of things that can go wrong, so you can get actionable insights and address them all individually. So they're maybe fairly simple in comparison to what I'm curious about, which is, I think there's been a lot recently about how do you, could these methods be applied, and is there an interest in doing that to increase lifespan?
So it depends on which population you talk to. So there are segments of the US population, older, well-off people that are very enthusiastic about this. I think one of the real constraints is the fact that the phenomic analyses cost roughly $5,000 per year, and they're not paid for by health insurance at this point in time. That's why the million-person project is critical. One, because it'll absolutely validate why health insurance should be paying these costs. But two, the program will drive down the cost of phenomics in the same way the Genome Project drove down by 7 orders of magnitude the cost of genomics. And if you can go from $5,000 to $5, that is something almost no one would object to.
Thanks.
Mm-hmm. Yes.
Hello. First of all, thank you for that lecture. That was a great talk. My name is Natalie Coles, and I published with you with my late husband, Dr. Stephen Coles, on supercentenarians at Stanford. And he passed away in 2014, and it looks like I haven't seen many people doing these studies anymore. So in an effort to try and continue sampling them, I wanted to come to your lecture, and I remember that you had donated 10 slots at that time. I feel like we've made so many advancements in the past 10 years, and so now that I'm trying to reignite this sampling, I was wondering what your opinion is about making them their own cohort, because part of the problem with the publications back then is we only had about 10 to 14 samples. And I've heard so much talk about age reversal, and I was thinking it would be great to use this particular cohort as maybe one of the populations that we use some of these studies with, age reversal. And I guess, so my two questions is, one of your P4, are you interested in participating in that again? And is there opportunity for me to discuss that with you after the talk? And two, I'm just curious what your thoughts are. I was thinking last night, I thought, if we were to be able to test again on supercentenarians, and let's say we have this assumption that they have a slower rate of biological aging, I was curious if you think that even if there is possibility of reversal, do you think that on a cohort like that, that their reversal would also be slower? And if not, maybe that's something interesting to discuss and continue sampling.
So let me tell you, I think the study of centenarians would be terrific. We studied, I think, 18 centenarians that were 110 or older, I don't know, maybe
Supercentenarians.
15 years ago, and really found some fascinating results. But
Right.
the criticism at the time was it wasn't a large enough sample to be statistically significant anyway. So that's one thing you have to worry about. But what has changed fundamentally is this: with these very dense data approaches to patients, you cut down by orders of magnitude the number of people you need. And the reason for that is very simple. People are worried if you have 10,000 measurements and they're all independent, you'll never be able to do hypothesis testing to see if they're independent. The 10,000 measurements, if you think in terms of systems biology, reduce into 300 biological networks, and that reduces the dimensionality of the data enormously. And I'm arguing, and we're doing the power calculations now, they're complicated, but my gut feeling is if we could even get a few hundred, we would have more than enough to be able to deduce very strong kind of conclusions. There is a longevity grant out of NIH, a subset of which is still studying centenarians, and I don't know exactly where that is at this point in time, but none of them are studying it with the deep data density, and that's what's critical for this population, because you're not going to get 30,000 of them.
Right.
But if all of a sudden you can do with 200 or 100, that's a different ballpark. So sure, we can talk. I do know about your work and everything, and I think it's great.
Wonderful. I'm scheduled to go next month to visit the world's oldest woman because the University of Spain wants to do some collaborations with testings and people here in the United States as well. So yes, I definitely want to talk to you if you might be open to doing that again. Thank you.
Mm-hmm. One more question, unfortunately.
Hi, Dr. Hood. I wanted to say very much I enjoyed your talk, and thank you very much for delivering it. My name is Noah Rahman. I'm class of 2009. And I guess my question is, there have been quite a few companies in Silicon Valley kind of commercializing machine learning models of RNA conformation with, the huge vistas of data and maybe and it's not Google, by the way, as far as I know. With the huge vistas of data and, tie up with Google in front of you, how do you envision utilizing these sorts of technologies, which are basically here for, deployment in your approach?
Right. Yeah, so what all of those companies lack totally are data, okay? And I mean dense phenomic data. And we will have that once we set this project up and get it going. But I think the other thing they lack is an understanding of how important it is to take these hyperscale AI systems like GPT-4 and actually properly educate them, and don't improperly educate them, because improper education leads to false answers and false conclusions and stuff. So what we're doing right now is together with Google, we're going to be using Google knowledge maps, knowledge graphs, together with Spoke Knowledge Graph from UCSF. We're creating, as I said earlier, a digital twin for diabetes, and we're going to put the knowledge graph plus, for the diabetes, you put in from the top the physiology and the mechanistic information, from the bottom the biochemistry and the omics and the clinical chemistries. And then we're going to feed in all the data from the Airvale population. And this digital twin will be absolutely the exact right thing to educate hyperscale AI with. And then you can come, as I argued, and there are arguments about in the future, maybe we won't just have to settle for statistical inferences about complicated data sets. Maybe we can make causal inferences. And of course, that's a point of debate, obviously.
Thank you very much, sir.